Okay, so just to review, right? Light comes in, electromagnetic radiation in the narrow range that correlates with visible light. It gets bent a little bit by the cornea. It works its way through the pupil, which can be dilated and constricted to let in the appropriate amount of light. It works its way, through the lens where it can be bent, and you have the ability to sort of stretch and, scrunch up that lens to change, the focal distance, and you can have it then, focused on mostly the fovea, right? That divot in the back there where you've got very, very tightly packed cone cells, those typically three, distinct photopigment segregated, short wavelength, which correlates with blue perceptual experience, medium wavelength, which correlates with green, and the L type or the long wavelength, which correlates with red. And remember, they're at the very back of the retina, with their outer segments with the membranous discs, where the photopigments are located, which absorb the light, which generates a change in the shape, which sets off a biochemical cascade, which we're going to see is going to inhibit the cell and prevent further release of neurotransmitter that's associated with the dark current, because typically these photoreceptors release neurotransmitter. Paradoxically, it's called the paradoxical dark current in the dark. And so you're getting the right amount of light in, so some are shut down and removing inhibition on output, and some are actually firing and inhibiting the bipolar cells and inhibiting output. But you're going to get a very complicated sort of pattern of output via the bipolar cell to the ganglion cell. And, that's to be complicated by the horizontal cells and the amacrine cells, which are distributing and, making those synapses more complex. And then you're going to get a pattern of output that works its way out through this optic disc right here where the axons sort of coalesce. And then you get a great big optic nerve carrying all the information that's coming in from that eye. And it's going to start working its way back into the rest of the central nervous system. The retina, by the way, is considered to be part of the CNS, part of the central nervous system. So when you look into somebody's pupils, you're looking right into their central nervous system. And then this is, a brain model. Your eyeballs are here and here. And then you can sort of see these little projections. Like here's one optic nerve coming from this eye. Here's another optic nerve coming from that eye. And then you're going to see there's a crossing called the chiasm. And we're going to see that everything, when I'm looking straight ahead, to the right in visual space, this is called the right, for me, visual hemifield, half of the visual field, the entire visual field. Everything on the right side that's coming in, you're going to see it's going to actually come in, both this temporal, or lateral retina over here in this eye, and it's going to come in the nasal retina over here on this side. Everything on the right side here, though, has to end up past that chiasm or crossing of the optic nerves on the contralateral side, the opposite side of the brain. So we're going to see that, for something that's out over here that I'm looking at, right, when I'm looking straight ahead, the reflected electromagnetic radiation is going to work its way in to the lateral retina here. And you'll note that this has already gone to the contralateral side of my brain. It's the eye on the opposite side now, the contralateral side. So when that portion of the optic nerve makes its way to the chiasm, right, it stays on that same side because it already decussated, right, crossed in space. But the stuff that's over here that gets reflected on the nasal retina or the medial retina of this eye, on the ipsilateral side, when those projections go back to the chiasm, they do need to decussate and cross at the chiasm. So the optic nerve we're going to see is carrying information from just a single eye, which will include information that's coming from both sides of visual space. But at the chiasm, we're going to cross the stuff that had not already decussated or crossed in space. And so post-chiasm, we're going to see we've got these, what are called the optic tracts. So further back, past the chiasm, those cables that are coming out of the chiasm, we have now fully segregated everything coming from the right side of visual space over onto the left optic tract, and everything on the left side of space over onto the right optic tract. And then from there, about 90% of the projections of these optic tracts, these are the axons of the ganglion cells still that are coming from, the ganglion cells in the retinas, are going to synapse with the thalamus. And remember, the thalamus is a critically important, central medial brain structure that is, receiving specific lines of afferent input, all different forms of afferent input, all different forms of sensory input, except, of course, olfaction, which is direct, privileged access into the brain. The lateral geniculate nucleus is the specific thalamic nucleus that processes that incoming visual information from the optic nerve, through the chiasm and then through the tract. The tracts are going to, work their way into the thalamus into something called the lateral geniculate nucleus and make synaptic connections there, bringing visual information in. And the LGN, or the lateral geniculate nucleus, is called that because genu is actually Latin for knee, and a geniculate is a little knee. And, actually, this is where, let's take a look, part of this brain off here. Take a hemisphere off. And so here we've got the brain stem. The thalamus is right here. Then back here, further, a little more lateral this way, right, is going to be a little bump. There's actually two little bumps. So the thalamus actually looks as if it's kind of kneeling, or at least it looked like that to early anatomists. So they propose that it looks like it's kneeling on little knees. And the bump that is more lateral, the lateral little knee, or the lateral geniculate nucleus, is going to be processing that visual information coming from the eyes. The medial geniculate nucleus, the knee that's a little closer to the midline, we're going to see is going to be processing auditory information from the ears. And then from the lateral geniculate nucleus, which is the thalamic nucleus again, that receives the incoming visual information from the eyes, you're going to see axons sort of radiate out towards the back, radiate in the white matter, below here, and then form synaptic connections in cortex. And this area right in here, in the midline of the occipital lobe, there's a fissure here called the calcerine fissure. This area right around here is primary visual cortex, or V1. And this is the first initial arrival site for visual information, in cortex, coming initially from the eyes.